Yes, sir.
Okay, fine. This type of colonies we call it a Drotman colonies, and this is ambonate colony. At the center there is a raised surface. Now, if you ask me why it happened, I don't have any answer. It is the nature of that organisms, they form this type of colonies. So at your level, I cannot explain it, why it happened. But this is a characteristic. It is something like why I am of this much size and shape. It's because I'm like this only. So for bacteria also, they produces this type of characteristic colony. And by studying these colony characteristics, we get a lot of information about the organism, and we almost reach near to the bacteria. In certain case, like here in this picture, you can see several different types. This is one round entire colony, and this is one large colony, flat type with ambonate. The center is raised. And this is a totally flat type, very large colony, almost a few millimeter. Okay, the surface is rough. So these are the things that, looking at these pictures, what I can say that in this particular culture plate, we are having 1, 2, 3, 4, almost 4 different type of organism. So if your clinical sample contain 4 different type of organism, and if you inoculate on the surface of the agar and grow and incubate them, then we are going to observe the colonies like this way. Now, very carefully, without touching any other colony, with the inoculation loop, if you touch this colony and put it in another plate, you will get the pure culture of this bacteria. So in the 4 different culture plate, you just touch this colony, and go on putting into 4 different culture plate, you will get the pure colony. So this is the process of isolation, the organism in the pure culture. Okay. So in subsequent classes here, we'll discuss more about this individual species and the disease caused by them and their identification and colony characteristics. More in details, we'll discuss it. But in this class, I just want to highlight it that this way, these are the things that we generally observe. Okay, this is in case of Haemophilus influenzae, and that is associated with respiratory tract infections in human, more particularly. And similarly, in animal, we have the Glyceria parasuis that causes respiratory tract infection in pig, and some systemic infections. So they produces this kind of watery colonies. The colonies look like a drop of water on the surface of the agar. Okay. This is another example of a Streptococcus organism which produces very pinpoint colonies. These are the colonies, very small type colonies in the blood agar media, and they're basically like alpha hemolytic. This is another clear close capture of some bacterial colony. We can see they are glistening type colony. Okay, glistening is their shining. The light is reflecting on the surface. So we have certain organisms which are dull in nature, means they are not reflecting, not shining. Okay, even if you look against this different angle light, then you won't see any glistening or shining. And then this is one characteristic that we can consider. If you know that your target organism is having these characteristics, then obviously we can think of isolating the organism. Yeah, this is another very cultural characteristics of a particular organism. This is the organism what we call as the Proteus species. So this Proteus, they always produces swarming colonies, just like the sea waves. You can see from our center point, their cultural characteristics, they grow like waves, water waves, and this is what we call as the swarming colony. I mean, they will use a spot inoculate into a culture plate, and gradually they will spread it to the periphery and this kind of growth. And now, if you just look at these characteristics in a culture plate, we don't need any other identification system. Straight away, you can say, yeah, it is that. Proteus species. Here it is, as you know, the Yersinia pestis that causes the plague, human. This is one of the most pathogenic organisms causing severe septicemia. So their colonies are sticky in nature. You can just see it here. If you try to touch with any inoculation stick, then they will attach it just like a rope. They will attach it. They produce this kind of sticky colonies. So these kind of characteristics are specific for certain organisms. So when we target for isolation of Yersinia pestis in the blood agar, obviously we look for this characteristic, whether it is present or not. If it is present, then we can suspect that these colonies might be because of Yersinia pestis. But remember, these are all colonic characteristics. Blindly, you cannot identify them and to genus and species level, not possible. But we can study and we can correlate with our target organism. Many times I'm talking about the target organism. Target organism means when some clinical sample specimen reaches to our laboratory for diagnosis purpose, then we take the history of that particular case, and then we suspect that it might be because of those organisms. So how we confirm is that we assume some target organisms and we try to explore those target organisms in the culture plate, and then accordingly we identify them. So this is a picture of Staphylococcus aureus. So this bacteria always produces a pigmented colony. That's why this particular species name Aureus. Aureus comes from the word aura. Aura means gold. They produces golden yellow colony. It is a pigment produced by the organism in the culture plate. Okay, so these are the golden yellow colonies produced by Staphylococcus organism. So they produce round, entire, circular, smooth colonies on the surface of the agar. And this particular media is a blood agar media, as you know. The last class we discussed it. When we incorporate animal blood, aseptically collected animal blood at the rate of 5 to 10% to molten agar rows, then we get the blood agar media. Okay, so in the blood agar media, when the bacteria they grow, if they have the potential to produce some toxins like hemolysin, is a common toxin, they can cause the hemolysis of RBCs. So here in this picture, you can see two different types of hemolysis. One is totally hemolyzed, which become very clear surrounding colony. We call it a complete hemolysis. Whereas outside of this complete one is a large or bigger zone of partial hemolysis. So these things we'll discuss later on when we talk about the Staphylococcus disease. What is alpha hemolysis, beta hemolysis? But at this stage, try to understand that how these colonic characteristics are studied. Okay, so when you target for Staphylococcus aureus, obviously we will look for its color formation. So this is another that causes the disease strangling horse, Streptococcus equi. They always produces or glistening type mucoid colonies. This type of colonies are also called as Mccoy colony, as if a drop of mucus is fallen on the surface of the agar. We call them as the mucoid colony. Okay, so when you know the colonic characteristics of the target organism and we try to correlate whether it is same or not in the culture plate of the strict plate method of inoculation and incubation, so thereby we can go almost, we can reach near to the organism. Now in this particular picture, you can see several different type of bacterial colonies. Like here it is a glistening type colony. Here it will be rough type colony, dull type. It's not glistening. Light is not reflected by these colonies. Whereas here you can see the very clear reflections. Sometimes we call them as the translucent colonies. Okay. Here also the same things here. They are glistening colonies, and in the blood agar they are showing some sort of hemolytic reaction. Here it is just like a water drop, dew drop like colonies. They are producing on the surface of the agar. So These are all different type of bacteria, and they're having their unique colony characteristics. Look at this picture here. You can see this the surface is not smooth, as if some sand-like particles are present on its surface. So we call them as a rough surface colony, and comparatively they are much bigger in size. This organism doesn't producing any hemolytic reactions, whereas they are producing prominent hemolytic reactions. So these are the things that we observe, and this is a very clear picture about the streak plate method of inoculation. If you can recollect, this area we call it as a pool. Okay, first we smear in one area, as we have seen in the previous class video. Then from the yeah, can you see the slide now?
Yes, sir.
Okay, because of power interruptions, the internet get delayed. Again, I have connected. Okay. You can see the slide properly, I guess. Yeah, can you just help me? You can see the slide, and can you hear me?
Yes, sir.
Okay. Yeah. So these are some other characteristics, as I said, about the margin or age of colony, as we have seen in the previous: entire, lobate, undulate, crenated, readily striated. Of course, all the time it is not possible for us to remember each and every bacteria and their characteristics, but whenever we go for isolation, we study freshly about their colony characteristics, and we try to explore those characteristics into the culture plate. Okay, then now you can understand about the smooth, the rough. Okay, and like these are the rough colony, these are smooth colonies. Okay, this is another rough colony, this is a smooth colony, Mccoy type colony. Then even the opacity, some the bacterial colony, the light passes through it. We call them transparent. Somewhere the light is not passes completely, translucent, and there are many colonies which are opaque in nature. Okay, so these are also the colonic characteristics, right? From the beginning, what we talk about the size of the colony, then the shape of the colony, then the elevation of the colony, then the surface contour, margin, opacity. These are different colony characteristics. Let us see here. In this culture plate, they have a lot of bacteria, and here in this area also a lot of bacteria are present, but you can look through it. That means these colonies are transparent colony. The light passes through this particular colony. Just think about the other hand. Here it cannot, light cannot pass through it. These are opaque colony. Okay, so the transparent colony, pinpoint. Okay, just like a water drop, and here the large type colony and opaque. These are some characteristics that we consider for identification purpose. Then coming to another characteristic called pigmentation. As I said about the Staphylococcus aureus, they produces golden yellow colonies, and looking at this pigment, it helps us in identification of the bacteria. So similarly, we had some organisms where the specific pigment formations can be seen, and that helps us in identification purpose. Then again, the consistency, there are certain bacterial colony which is butyrous in nature, just like the feeling of taking some butter from a block of butter. Okay, with a spoon, if you can easily take out the butter, we call as a vitreous consistency, whereas there are certain which they always produces very sticky type colony. Even certain organism they produces powdery colony, like if you try to pick up with the inoculation loop, the whole colony breaks. Okay, so that's a consistency, and lastly it is the odor. Odor means of course not all bacteria. There are certain bacteria which produces very peculiar odor, and then when we get that odor from the culture plate, we suspect the presence of that organism. So not only all the visual things, even the odor also plays a role in identification of the bacteria. So as such, these are the different type of characteristics that we study. If we closely, shortly go through once again, then you'll understand. After inoculation, the bacteria in the solid medium, we study the shape, size, elevation. Then we study the merging or edge of the colony surface, then opacity. Okay. Then we talk about the pigmentation, consistency, odor. So these are the things that we recorded while studying a culture plate. Let us come to this particular culture plate. Straight away, you can say they are producing golden yellow color colony, pigmented colonies, and these colonies are almost 1 millimeter in size, and they are opaque, like cannot pass through it, on their smooth, round edge. So we correlate with our target organism. If it is matching, then might be our target. Similar way, this one is the plate of Staphylococcus aureus. It's a common pathogen causing the disease mastitis, and most of the times we suffer from acne and some abscesses in skin and the face. And this is the organism which is responsible for that. And this is another species of Staphylococcus. It is Staphylococcus aureus. They produces white colony instead of pigmented colony. They doesn't produce golden yellow, rather they produces white pigment. So in the blood of the media, we can detect it. And these are the Staphylococcus aureus. Okay, so after studying the colonies, we can get a rough idea about this organism. Okay, so next important part is to see the morphology of bacteria. Some of the physical characteristics like motility also an important criteria in identification of that particular bacteria. So in the morphology, we go for the staining, different type of staining that you have learned, a differential staining like acid fast staining, ground staining, or maybe simple staining, or maybe we call it spore staining. These things we perform, and then we record the observations. So we basically study the shape, whether it is a coccus or a bacilli, then the size, how much it is smaller size or the bigger size. Then what about the sides, where the sides are parallels or they're elevating or bulging. Okay, ends of the bacteria, whether they're sharp edge or they're round edge, etc., etc., including a phenomenon what we call as the pleomorphism. Please note it down. This is important for you. Pleomorphism. This particular word, Pleomorphism. You understand this is a common English word. Pleomorphisms. It is a phenomenon we seen among certain bacteria. They change their shape and size in different environmental conditions. We call them as pleomorphic bacteria. For example, the bacteria, they may appear as a rough-shaped bacteria in certain culture media or certain cultural condition, but at the same time, they may appear as round in animal tissues. So we call them as pleomorphic bacteria. The bacteria which change the morphology depending on their situation, environment, and the culture media. Of course, not all. There are certain bacteria which are pleomorphic in nature. So if you know that your target organism is a pleomorphic bacteria, then we have to consider the different shape and size of that organism. Okay. Then presence of capsule, flagella, and you know the motility, as we learned in our previous class about the hanging drop method. through which we can find out whether the bacteria is motile or non-motile and the phase contrast microscope. So these things we have to carry out after streaking the bacteria in the culture plate, and we get the pure culture and we study all these different characteristics. Spores is another important structures. As you know, not all bacteria, there are certain bacteria which can transform into the most resistant form of life, what we call as the endospores. Okay, these bacterial spores can survive for quite a long period, and they are very rough and tough. And remember again, not all, and whenever they get the opportunity, they can germinate into the vegetative form and can produce very, very severe disease in animal and human. So if you know that your target organism is a spore forming bacteria, then try to see the spores by spore staining method, how the spores look like. Is it central, terminal, or bulging, non-bulging? I guess these things you already studied in the theory. Subsequent slide we are going to see commonly. This is the process of identification of bacteria. Okay, practically you need to learn it in the theory class, practical classes. Let us randomly see some morphology and staining characteristics of bacteria. These are Streptococcus, means coccus in long chain. As you can see, they are gram-positive bacteria. They are taking the violet color of crystal violet, and they are coccus in chain, long chain. Here it is another long chain. This is the most notorious bacteria that are causing the sore throat in human. And in animal, most of the time it is associated with the disease mastitis. They cause inflammation of the udder. So these are the Streptococcus, coccus in long chain, and all are taking the gram-positive staining. Okay. These are the Staphylococcus. They are also coccus, similar to the Streptococcus, but these coccus, they are arranged in cluster, just like the bunches of grapes. Okay, so we call them as the Staphylococcus, coccus which remain in cluster. This is another picture. You can see a close view of how this round organism, almost the size of 1 micrometer in diameter, because they are round. They aggregate just like a cluster. That's why they are called as the Staphylococcus. So from the culture plates, if you can demonstrate it, it is quite easier for us to identify the bacteria. This is a very pathogenic bacteria of animal species Clostridium jovi that causes the disease black quarter. This causes a necrotic myositis conditions. The whole muscle scale get rotten because of this bacterial infection. And they are under the group of Clostridium. They are anaerobic bacteria, and they produce spores. And the spores, they are lemon or almond shaped, bulging out of the exosporium, and basically they present in the subterminal region. So if you guess that the case might be because of black quarter infections, then obviously we observe this is an impression smear taken from the muscles, rotten muscles, and we can see this bacteria that help us in identification. And this pictures is very familiar to you, I hope, and that these are the Clostridium tetani that causes the disease tetanus. Okay, they are also Clostridia, but they produces terminal round bulging spore, look like a drumstick. Also, we call it as a drumstick bacilli. Okay, it is a synonym of Clostridium tetani. Some literature they say is a tennis racket also. So many ways we can explain it. This is only because of the terminal round spores, bulging spores present at one end of the bacilli, And these spores are highly resistant to the environment, and entry of the spore to injured tissues lead to development of the disease tetanus. As you know, tetanus is a terminally fatal disease in man and animal. So to identify these bacteria, we can demonstrate these structures. Even from the culture plate, we can see this structure. This is another Clostridium tetani, tennis racket shape. You can see smear taken from some culture plate. Then, yeah, this is a schematic diagram showing different types of terminal, then terminal round, terminal oval. Then this is subterminal, and this is somewhat central. So depending on the location of the spores, we can identify the bacteria. Like we know that the Clostridium tetani produces terminal round bulging spores. So if your bacterial smear shows that the bacteria is presenting some central spore, it cannot be Clostridium tetani. So these are some good characteristics through which we can identify the bacteria. And here it is showing some porphin genes which are producing subterminal spores. These are green color spores. It is the malachite green staining they perform it, and exosporium is taking the purple fuchsin color. So likewise we can demonstrate it. So this is another criteria that we consider. Yeah, this picture is a very clear-cut picture of a septicemic disease that occur in cattle and buffalo very frequently in our country. It's called as the hemorrhagic septicemia. The disease is called as the hemorrhagic septicemia, and it is caused by the organism Pashurellum maltosida. So these bacteria, and these are the RBCs. These are the RBCs from the blood smear. Septicemia, as you understand, the bacteria, when they circulate in the blood, we call it as a septicemia or bacteremia. So bacteria, along with its toxins, when it circulates, we call it as a septicemia. So at this particular stage, you can see the bipolar nature of the bacteria. These are some bipolar. These are basically gram-negative, but this is not a gram-stained smear. This is a Giemsa stained smear, where both the end of the bacteria, they are taking intense color. We call it as a bipolar staining reaction. Okay, so this picture, if you can demonstrate, then immediately you can say it might be because of Pashurellum maltosida. So Sculptural Characteristics, how pass in identification. This is a picture of, again, the Bacillus anthracis that causes the most lethal disease called as the anthrax in animal. Human also suffer from this infection. So they produce relars type bacilli, and the end of the bacilli is very sharp edge, truncated, what we call. It's not round. The difference between the Bacillus anthracis and the Clostridium is that Clostridium species, the ends are round. They're round, whereas in case of the Bacillus anthracis, it is truncated, sharp edge, just like a card box. These are some of the synonyms that we use it. Truncated end, sharp edge end, the characteristics of this bacteria, and also they produces the endospores. Of course, in these pictures, the endospores are not clearly visible. Just a glimpse I'm giving you. There is another bacteria called the Corynebacterium species, and some people, they describe that they're under the microscopic field, the Corynebacterium look like some Chinese script. So one of the synonyms of the bacteria is the Chinese letter arrangement. The arrangement is called as the Chinese letter arrangement, as if these are some of the Chinese script or Japanese script written somewhere. But in all textbooks, you'll find this commonly described it as Chinese letter alphabet arrangement. It is related to Corynebacterium. Basically, what happens is bacteria, when they grow and divide, the daughter cell usually do not get detached from the parental cell easily. So from the second daughter cell, when the other cells get divided, then they remain in cluster, and that give rise to shape look like the Chinese scripts. So it's a synonym of Corynebacterium. This is another bacteria called as the Fusobacterium. Okay, so they always produces filamentous growth, like just like a hair top. At certain point, people were confused with the fungi, but later on found that they are pure bacteria. They are not fungi, and they are belonging to an aerobic group of gram-negative bacteria called as the Fusobacterium. They're commonly associated with condition what we call as the food rot conditions. When our animals are stranded in muddy floors, then this bacteria, they produces some necrotic lesions in the hoof. We call it the foot rot condition. So we'll talk about this different diseases at a later point. Just for your idea, what I'm telling you is that in the identification of the bacteria, we start with inoculating the bacteria, the pure culture of the bacteria in the liquid broth media as well as in the solid culture media, and we study the colony characteristics. So in colony characteristics, whatever the things we discuss it about the shape, size, elevation, then their ages and appearance, pigment protections, odor, everything we considered it, and we make a list, and then we try to match with our target organisms for identification. So that help us almost reaching to our target organism. But after that, we need to go for the staining reactions. So staining, the regular differential staining, we perform it, and some special staining, even the spore staining. Then we generate a lot of information about this organism, including the motility. We study it, motile or non-motile. And then almost we can identify the genus of the organisms up to that bird. But if you want to identify the species of bacteria, it is not possible on the basis of this cultural characteristics and on the basis of staining reaction. You cannot identify. So within the same, suppose the Staphylococcus, we had a different type of species. Okay, the Passerella, we are having different type of species. The Salmonella, we are having different type of species. So within the same genus, if many species, if you have to identify, then we have to go for the next stage of bacterial identification, and that is on the basis of the biochemical test. So what we have decided that at this point we will not discuss about the biochemical test for identification of bacteria, and it will be dealt with the chapter when we start discussing about the systemic infections, bacterial infections, and when we start talking about the individual disease producing bacterial genus and species, then this will be more relevant. So this part we'll discuss at a later point of time. So the biochemical test, why it is needed, let me tell you. At certain points, the species of bacteria, they differ from one another in slight characteristics. Minor characteristic differences are present, and this can be explored with the help of different biochemical tests. So, and these are different biochemical tests that we'll discuss at a later part. Okay, so coming to the last part of this general bacteriology, we had a topic on antibiotic sensitivity assay. As you know, this is a very helpful tool for the veterinarians or the clinician to select the most appropriate antibiotic to be used against a particular disease-causing agent. Okay, as you know, these days again a lot of controversies and recommendations are coming. Which antiviral we should take it, which not, which is effective, etc., etc. Similarly, in case of the bacterial infection, as you know, the antibiotics is the solutions. We had different types of antibiotics, and these antibiotics, because of their prolonged use for treatment in animals and human, they are again very clever creatures. They are start developing some sort of resistance against this antibiotic, and antibiotic resistance become a big issue on today's date. And you just think about that if the bacteria could able to generate the resistance against most of the antibiotic available in the market, then what next? We will be handicapped for treatment against the bacterial infection. And in fact, it is happening. In fact, it is happening. All of you must be aware about this drug-resistant phenomenon appearing at a very rapid rate in the population, and it become a major concern for human health as well as animal health. So in 50s, when the first antibiotic was started with penicillin, and then later on we had a lot of different type of antibiotics there in market, and people are using this antibiotic for treatment purpose. And now gradually, after use of this antibiotic for quite a long period, we have observed that the bacteria are changing their genetic material and they are becoming resistant to these antibiotics. So this antibiotic is no more effective against the bacteria. So newer and newer antibiotics people are sourcing, and they're in the market, and it become a continuous process. When new antibiotic come again, the bacteria to transform themselves and try to become resistant against it, because this is another struggle for the bacteria to survive in the nature. So this tug of war is going on. And then coming to this particular point, like how we can identify that a particular antibiotic is resistant or sensitive against a particular bacteria. So we can perform a very small test called as the disk diffusion method, and it was developed by Kirby and Boer first time, and then that is very useful, and all laboratory they're using this technique for identifying the sensitivity and resistant pattern of a bacteria against the common antibiotics. So the basic principle in this technique is that it's a very simple technique. First, we take an agar media, and then we spray the pure culture of the bacteria on its surface. If we keep this plate as such, what will happen? The bacteria will grow and they will form a lawn of bacteria, as you can see it here. So we don't incubate without adding antibiotics. So what we do, we spray the bacteria on its surface, agar surface, and then we place immediately some antibiotics which are impregnated with the paper disc. These are some round paper discs which are soaked in a definite concentration of different type of antibiotics, and we place them at a regular interval on the surface, and then we incubate the plate. What will happen? This antibiotic during incubation, they will diffuse from this disc, paper disc, to the surrounding media. And if the bacteria are susceptible to this antibiotic, they cannot grow in this area where the antibiotic get diffused. If the bacteria is resistant, then what will happen? There will be uniform growth of the bacteria everywhere, covering this whole disc. There will be no clearing of the area. This is what we call as a zone of inhibition. Here, no bacterial growth could take place because of the antibiotic. If suppose this antibiotic is the bacteria is resistant against this antibiotic, then you cannot see this zone of inhibition. Rather, they will uniformly grow just like this, and you cannot see the zone of inhibition. So there is a process of measuring the diameter of zone of inhibition, and then we compare with the standard search to declare that this bacteria is sensitive or resistant against that antibiotic. So this is a very simple method, and so overnight we can get the results. And getting these results, the clinician can select the specific antibiotic for treatment purpose. Okay, this is called as the Kirby-Brewer disc diffusion method. So this is another practical. Always we give demonstration to the student. Hopefully after your return we'll do it. So I just downloaded a small video from the YouTube. Let us see, and this will be the last part of today's discussion. So here and they will be describing how this diffusion method is performed. Okay, so in this particular assay, we need the culture plate, sterile culture plate. We need a culture and pure culture. Remember, always we need a pure culture of bacteria and some inoculation swab, and then we need the antibiotic disease. Okay, so we use some small forceps to pick up the antibiotic disc and other material for sanitization. So this is the culture plate, maybe the antibiotic test sensitivity agar media or Muller-Hinton media, or even we can use a neutron agar plate. Okay, thing is that it should be a clear plate without having any indicator. Okay, then we take a swab and then culture in the broad culture, but this culture should be a pure culture. Okay, so near a spirit lamp, we open it. These are the aseptic techniques. We follow it, and then we take the bacterial culture through the swab, and then we smear it on the surface of agar media. Can you see the video? Can you see the video?
Yes, sir.
Okay, fine. So here's the spreading all over the surface. It's not a strict plate method of inoculation. This is spray plate. He's spreading. We want that the entire part of the bacterial culture media surface spreaded uniformly with the bacteria, so that the bacteria can grow just like a lawn. And then he divided into 4 parts, and these are the different antibiotic dishes. So one by one, this antibiotic. These are paper dishes soaked in antibiotic solution. Okay, so every time he's sterilizing the forceps, and then he's picking up this antibiotic dish, and it's placing in the different location. Okay, so what will happen, because of the media surface is wet in nature, this will stick to the surface. Okay, and gradually this antibiotic will be diffusing out of the disc. Okay, and then if the bacteria is sensitive to the antibiotics, they cannot grow surrounding this diffused area. But if it is resistant, then the bacteria can grow. After that, we have to incubate it. These are different codes they are giving for antibiotics, like kanamycin, this penicillin, that gentamicin, etc. And then you can see this result. Okay, here what happened? This particular antibiotic, you can see a zone of inhibition. After 24 or so incubations, you can see this area, no bacterial growth can take place. So we can take the diameter, we can measure the diameter of zone of inhibition and compare it to standard SERT. Here also you can see a zone of inhibition. Against this antibiotic, this also you can see a zone of inhibition, but the diameter is too less in comparison to this and this antibiotic. So after comparing with the standard SERT, only we can say whether it is sensitive or resistant. If the zone of inhibition is more than the recommended one, then we can say it is sensitive. If it is less, then we declare it as a resistant. Okay. Here in this case, you can see this antibiotic. There is no zone of inhibition. Everywhere the bacteria has grown, just like a lawn or carpet growth has taken place. There is no zone of inhibition. That means the bacteria is totally resistant against this antibiotic. Probably it is the penicillin. Okay. So likewise, we measure the zone of inhibitions, and then we compare it with the standard SERT to identify and declare that bacteria is resistant or sensitive. So this was what I prepared for today's class, and from the next class we will start the immunology section. Okay, if you have any questions or any query, all the classes I'm requesting you can put the questions in the WhatsApp or maybe in my mail or in the class itself, so you can ask the question. so that I can clarify it. If you have any specific questions, you're most welcome. You can ask me, and if not, then thank you very much. I'll be winding up today's